Hello, my name is Christine McGowan. Today is January 26th, 2023, and I'm doing this for my cell biology class. Okay, so when talking about cells, you have two main kinds. You have eukaryotic and prokaryotic cells, and there are several main differences between the two. So I'll start with a prokaryotic cell. A prokaryotic cell is much smaller than a eukaryotic cell. It's approximately, as you can see in this picture, it's approximately 1 to 10 micrograms, or micrometers, I apologize, compared to a eukaryotic cell, which is about 10 to 100 micrometers. So a lot smaller. The prokaryotic cell is a lot different than a eukaryotic cell, meaning they're mostly bacteria. They have around them a capsule, and on the outside of the capsule are small appendages called pili or fimbriae, which help with cell-to-cell adhesion, which we'll see when we talk about the interaction between prokaryotic and eukaryotic cells. Inside of the capsule, they have a cell wall, which is really important distinction between animal cells and bacteria cells. In the prokaryotic cell wall, they have a cell wall made up of peptidoglycan, which is a bunch of sugars and peptide bonds. And depending how thick it is, it's either a gram-positive or gram-negative bacteria, which definitely impacts how you would treat or work against that bacteria cell. So the way different antibiotics work to get through that peptidoglycan cell wall. Now, there's also a flagellum, which can be present or not. It's used for propulsion for the prokaryotic cell, so to help it move. Inside the cell, you will have cytoplasm, but you'll notice there are no membrane-bound organelles at all, especially no nucleus. So instead of having a nucleus with DNA inside, there's a nucleoid region, which is basically just all of the cell's DNA that's coiled kind of tightly. But it's important to note that it's single-stranded. So ultimately, it does look all looped together, but it is just a single strand of DNA, which is really important when it comes to cell division and replication. The only other thing that the prokaryotic cell has are these little, I don't know if you can see them, the pink dots are ribosomes. The ribosomes are important because they help make protein. It is important to note that the ribosomes in a prokaryotic cell are different than the ribosomes in a eukaryotic cell. And that they make different proteins, which is very important because there are certain antibiotics that only work against the prokaryotic ribosomes. So they attack the subunit basis of the ribosome that's found in prokaryotic cells, not eukaryotic cells. So if you look in comparison then to a eukaryotic cell, there's a lot more going on. The picture I have here is of an animal cell. There's a few clear distinctions between this and a plant cell, which I will note. So the main difference is that there is no cell wall in this animal cell, just a plasma membrane, which is that prototypical phospholipid bilayer, which the prokaryotic cell does have a cell membrane, and it basically just allows, pushes up against that cell wall. So that would be the green in this picture is the plasma membrane. It just pushes up the cell against the cell wall. It allows the cell to keep its shape, but also that it's constantly full of fluid. So the plasma membrane helps to keep that in. It also works the same way as a eukaryotic cell in allowing permeability into and out of the cell. So again, the eukaryotic cell has this semi-permeable membrane that allows things to diffuse into and out of the cell. Additionally, you'll notice we have a nucleus. The nucleus is going to be home to all of the genetic material, which here is chromatin. Important to note in eukaryotic cells that it is double-stranded DNA, so the process of replicating takes a lot longer, and then cell division takes a lot longer. Inside of the nucleus is a darker circle called the nucleolus. This is where ribosomes are made. Outside of the nucleus, you'll have the endoplasmic reticulum, both the smooth and rough ER. Smooth ER is going to be the site of protein synthesis. Rough ER is going to have ribosomes attached, so it's going to be more readily for protein synthesis. You also have ribosomes throughout the cell that have moved from the nucleolus and rough ER. Down here you have the Golgi apparatus, which has to do with packaging things to move within the cell and then outside of the cell. Floating in everything, you will have cytoplasm. These lines represent the cytoskeleton. There's all sorts of different things that help to move the organelles within the cell and also help pull the cell apart during mitosis and meiosis. You have vacuoles, which can just contain food or can be getting ready to leave the cell. Lysosomes, which are important because they help in breaking cells down, either by phagocytosis, by the lysosome breaking down the cell itself, or by breaking down other things inside the cell that it no longer needs. A plant cell would have chloroplasts and a cell wall. Those are just the distinction that a cell would need to do photosynthesis. Other important factor is the mitochondria. The mitochondria is where ATP or energy is made for this cell. So when you're talking about cells and you're talking about that prokaryotic to eukaryotic interaction, basically they can be used for a lot of different ways. Sometimes the prokaryotic cell can be beneficial and help improve the junctions between the prokaryotic and the eukaryotic cell. So for instance, in the paper that I looked at, it was discussing the gut microflora compared to a person that has kidney disease and how typically the microflora is important for protecting the barrier and increasing the integrity of the junction between the epithelial cells within the intestines. When a person has kidney disease and the amount of urea in their bloodstream goes up, it causes the integrity in those junctions to separate, and then also leads to further kidney damage. The other thing to note is that they are attached by these pili, which can just attach to the outside of the cell. And then also, one of the important things about bacteria cells is they can talk to each other via signaling. And basically, the signaling mechanism can alert the cells to either keep growing and dividing, or to stop, or to start making a new protein, which is important because that's how cell growth or bacteria growth can happen. Again, note that eukaryotic cells undergo mitosis and meiosis, a much longer cell division and replication process, whereas a prokaryotic cell only undergoes binary fission, which is this growth and then separating, happens much quicker due to the single-stranded DNA. Thanks.